Welcome to Let's Talk Micro. Hello, dear listeners. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And you can always find Let's Talk Micro on Apple Podcasts, Spotify, Amazon Music, Goodpods. Wherever you listen to podcasts, you can find Let's Talk Micro. As far as social media, I am on Instagram, TikTok, and YouTube as Let's Talk Micro, on X as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and I have an email address which is letstalkmicro@outlook.com. So either via social media or via email, you can reach out with any feedback, any suggestions. So please go ahead and subscribe to the podcast, download episodes, and if the app allows you to do so, please leave a review. Thank you so much for the support, and I wanted to share with you this great milestone that Let's Talk Micro hit. Last week, we surpassed 100,000 downloads. So, oh my goodness. Thank you so much for making this possible. Thank you so much for supporting the podcast. I am so grateful. So please continue downloading episodes, share it with friends, co-workers. Let them know that Let's Talk Micro is out there, that it is a great podcast with great information. And I am always striving to bring you some great content that applies to your professional lives and to your knowledge. And Let's Talk Micro is a podcast that does not have any sponsorships or anything. And I always like to find great content, by going to different conferences, just such as, the American Society for Microbiology. I've been to ID Week, and I go to other ones to meet guests, to listen to talks and then find content, which is, I hear the talks and then I reach out to the speakers and they agree to do the podcast. So if you want to support the podcast in any form, in the show notes, there are two links, either via Venmo or via Ko-fi, Ko-fi. Those links, over there you can make a donation to the podcast so I can continue going to conferences and bringing great content to you. But anyway, I appreciate all the downloads and everything, so thank you so much. And of course, if you are looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. These are some great resources from Dr. Timothy Gauthier, a very passionate pharmacist. Well, today's episode is a little bit longer than normal, but it is some great content, so I hope you enjoy it. So I'm gonna be brief. If you haven't checked out the previous episode, please go ahead and do so. It was an episode about listeria, media, and testing. I go over some media used for listeria, which is pretty much used when it comes from listeria found in food items. I talk about Oxford, Palcom. I talk about chromogenic agar for listeria. So if you haven't checked out this episode, I invite you to please go ahead and download it and check it out. So I am definitely very excited to share today's episode with you because, as someone that loves microbiology, I wish that I had been, that I had learned it in high school, which I'm guessing like most of you in high school, you took biology. So I am so excited that this is being taught in schools. And in this episode, Meredith Townsend from Texas, she joins the podcast to talk about teaching microbiology in high school. And she breaks down her program, what she teaches, things like gram stains, antimicrobials. They do so much. And I love it. In the episode, I ask her if this is something that is being frequently taught in schools. She talks about that. As I say, she talks about what she teaches. She talks about how she evaluates the students. She also talks about her blog, which I'm including all her links on the show notes. But overall, I am so happy that this is being taught in schools. This is so much information, as I said over and over again. And maybe we start learning it in high school and it gives you a leg up in college. And not only that, it's, as you listen in the episode, not that many teachers are teaching this in high school. But she's doing so much. And she's so passionate, which I love it. She really cares about her students. She's really doing an amazing job. So shout out to Meredith Townsend. And thank you for taking the time to be in the episode and talk about this with me. So, great episode, great content. Let's go ahead and listen to it. So all this time that we have been talking micro on the podcast, right? We have talked about medical laboratory science programs. We have talked about microbiology in that sense. We had discussed articles. We have talked about antimicrobials. But what about microbiology in high school? We haven't talked about that yet. So this is a great topic. And as I find great content in social media, and that's how I met pretty much all the guests that I had on the podcast. And this time is no exception. I saw this teacher, and she was teaching microbiology in high school, and I thought, what a great idea to talk micro. So today with me, I have Meredith Townsend. Meredith, welcome to Let's Talk Micro.
Thank you for having me.
My pleasure. So I already told the audience your name. Can you do a quick introduction?
Yeah, so I am a high school biology and microbiology teacher in the DFW area in Texas. I am a mom of three kids, so two high schoolers and a middle schooler, and I'm also wife to a football coach, a college football coach. And so while teaching is a passion of mine, so is being a mom and chasing my kids around. So
Well, definitely welcome again. So let's start. How long have you been a teacher?
this will be my 16th year, and I've taught biology for 16 years. So I've taught a freshman, ninth grade. I've taught on level honors biology for quite some time. I've taught a little bit of IPC, and then this will be my third year teaching microbiology.
That is great. And you mentioned it, and when I went through school, right? So I took biology in high school, but I really started learning about microbiology once I was in college already, which had this been taught in high school before, right, it would have been so great. I mean, 'cause the moment that I started learning about microbiology, it was like a love at first sight, and I'm fully embracing it, and I'm so deep in it. And so this is great, but I don't think that this is routinely taught in schools, right? And if not, what made you decide to teach it?
Well, in Texas, where I am, we have what's called a CTE class, which is a career and technology class that the state of Texas TEA will fund if you have these in your school. They range from all sorts of different topics, like forensic science, for example. And so that is why our school currently has it. But you really don't see it taught very often. It's an expensive class. It has some unique features to it. Finding people who want to teach it is hard because it's not necessarily something that everybody took or even wants to teach. And so there is a small pocket of us. There's some all over the US as well. Interestingly enough, I started a Google group this summer. It's got about 35 high school microbiology teachers in it. And we meet over Google Meets. We have a shared Google Drive. But we did this, or I reached out for this because there's no curriculum out there. You have your textbook, but there really isn't just anything that has like lesson plans and things like that. And so I found a unique need to try to collaborate with people. And so there's about 35 of us, but we range all over the US, and there's quite a few of us in Texas. Um, but this course can range from a standard level course to an honors course. Some people teach it as a post AP course, which is how I teach mine. My kids have to actually take AP bio before they can take medical microbiology. And so, no, this is not routinely taught in schools. You really don't see a ton of us out there. Um, and to be quite honest, the reason I got thrown into it was kind of by accident. I'm the department chair as well at my school over science. And the lady who started the class unfortunately passed away. And when I got hired in, we had given it to another teacher who just really had other preps. She had two other preps already. And so I was just trying to, relieve some of her teaching load, and I took it on. And I have loved every minute of it. So I'm really glad that I was able to do that.
That is great. And yeah, that's something that I say that with social media and things like that, you can find so much information. And just like you created your group, and like you reached out, and then now there's people that they're doing the same thing, and sharing information. And that's one of the things that I do love. I mean, with all the negatives, right, that social media has, and but definitely when it comes to science and other things, it's just so great because a lot of people are willing to share their information. They're willing to help out. They want to share their articles, and they want to collaborate. They want to network. And it was such an eye-opening experience for me when I started this podcast and this journey. And it's so great. Um, Okay, so yeah, so let's talk about your class. So I'm thinking of microbiology in the terms of when I'm taking it as a medical lab scientist or regular student. So we typically, right, we do a lot of maybe some biochemical tests. We go over media, we plate organisms, we do smears. So how does it all work? I mean, what do you do in your class?
Well, that is a loaded question. we do we do a lot. So interestingly enough, when I get my kids in, even though they've taken honors bio, even though they've taken AP bio, they have very few microscope skills. Like like very few. I mean, they may have used it 3 times, 4 times possibly. And some people have taken, they took AP bio as a sophomore, and I'm not seeing them until they're seniors. And so there's this 2 year gap. And so honestly, we start with like just basic microscope skills and like what even is microbiology. Most of the kids took this class because maybe I taught them as a freshman and we just wanted to take a class again with each other, and actually have no idea what they're getting into. So it's kind of interesting. So we start with just a basic overview of the course. We do what's called diversity lab stations, and essentially they go through each major group of microbials. And so we do bacteria, archaea, protozoa, fungi, and they're learning to do wet mounts. They're learning to simple stain. They're learning how to just look at slides on oil immersion because they've never done that before. Um, and that takes like a week. Like you think, oh, that probably doesn't take very long. Oh no, it takes a good bit of time. So I have about 30 kids in each class. So 45-minute classes, 30 kids, 15 microscopes. And so you're trying to teach all of these skills to these pairs of kids. And my name is probably called out, 100 times a class period. Miss Townsend, I can't find this. Miss Townsend, I can't find this. And so for a week, it's just me running around trying to put out fires and help kids learn how to use a microscope. And um, it's probably their first time to really ever find anything. So we look at pond water. They do a little scraping of yeast. We literally just buy baker's yeast and I put it in water with some sugar and get it nice and frothy. And we look at that and they're like baffled that they can see these things, right? And so all these light bulbs go off. And I'm telling you, I know you said this, if you could have had this in high school, you would have found your passion. That's exactly what we're trying to do. Is we're trying to introduce all of these different topics to kids in a year. We cover a lot of things in a year, but just enough to kind of grab their attention and get them excited so that they can go on and maybe find a passion for that in college. So that's just the first week. We have they also get a credit for pathophys in my class, and so I also cover the immune system, and things like that at the beginning just to kind of get them going. We cover eukaryotic diversity, prokaryotic cell anatomy, metabolism, antibiotic resistance viruses, and what's the last one? Oh, epidemiology. So those are the units that we actually have that we go through. And I'm happy to talk about any of those units if you have, just the specific questions on what we do in those.
Well, I do, and before I get to that, yeah, I definitely agree with you. And when it comes to microscopy, that's something that even when in the MLS program that I teach, when you get them the first few days and getting used, because maybe it was maybe a year or two, after they took out general micro, which is a prerequisite to get into the MLS program. Something very interesting that I experienced was that during COVID, I eventually had a class that took their general micro online. So they hadn't worked with the microscope. So it was all doing videos and simulations. So that was extra challenging for them to get the hands on and working the slides and looking at things. And it requires time. I remember when I was going through and it took me a while to be able to look with both eyes. I don't know why. It's just like I used to, yeah, both oculars, I could only look at one of them. And then finally, eventually, after practice, and I just moving around and finally was able to see with both. So that was interesting. You mentioned antibiotics, antibiotic resistance, which is right such a heavy topic, and even in a medical lab sciences program, you touch on it, and there's so much information. That could be some classes by themselves, and it's constantly changing, and a lot of it you learn on the job. So I'm just curious about what do you cover in that area?
Yes, there's a lot. So Luckily, I do have a book. I have a textbook that sort of guides me. It's been really helpful in my journey of just like how deep do I need to cover things. It's a basic, it's a college level microbiology book, but it'd be like your first level. And so I tend to go with that first. So it's funny, we actually just finished it. That is what we've been doing. And so the first thing we do is first we cover what antibiotics are, because that alone is something that they don't understand. They know they've taken antibiotics. They know that it kills bacteria. Well, they think they know that. Sometimes they think it kills viruses. They have this unintentional knowledge, I think, of what it really is. So we first just cover what antibiotics are. I actually give them a project to do this. So I assign each of them a different antibiotic, and we go over resistance mechanism, or first of all, we go over their mechanism of action. How do they work? What class are they in? Where did they come from? Like, did they come from a bacteria? Did they come from a fungi? Did they come from a laboratory? They were shocked to know that a lot of antibiotics were derived from bacteria themselves. Like, that was what do you mean? It kills I don't understand. And so, where do we find them at? We talk about, penicillin a lot because that was kind of the original, right? Like, that's we go over those. And they do a project where they have to create their antibiotic, and this will maybe sound cheesy if you're not a high school teacher, but they basically make it into a superhero, and they have to find a way to kill the bacteria, and they have to make like that little children's book. And the whole point of it is for them to take something that's a really big concept and break it down into small pieces so that they could teach it to somebody else. That's my main goal, is for them to have a basic understanding of how they work. And so we start with that because that's a big one, right? Like, just how does that work? Then I found a simulation that basically you use beads and dice, and it's supposed to simulate what happens if you stop taking your antibiotics. And so the red beads are super resistant, the yellow beads are kind of resistant, the green are not, and they roll a dice, and the dice tell them whether they took their antibiotics or not. If they did, they kill off the green. If all the green died, they can kill the yellow, so on and so forth. And essentially what they do is they look at how long it would take if you skipped taking antibiotics, and that it would take longer than what your prescription's probably even for, because now they've multiplied and they're only going to kill a certain amount at a time. And that's really just for them to see kind of the idea behind it. So that's what we do to kind of introduce it. I love the Harvard video on YouTube where they make that giant agar plate. I'm sure you've seen it. And they put like different, what's the word? Different amounts of antibiotics. So they start with like 0%, 1%, 10%, 1,000%. And it shows that I think in 11 days, it takes the E. coli to completely overcome the antibiotics and become resistant and get all the way to the middle of it. And I show them that, and they are just like, what? Like, they can't even believe it. They think it's the most fascinating thing in the entire world. So we do that. We do watch a documentary, I believe that PBS put out, about just government funding and how we're not funding antibiotic research, and that's part of the problem we have. And then we talk about like how in other countries you can just walk up and get whatever antibiotic you need, and how that is contributing. So we talk about all of these different like social things, right, that have caused this kind of place that we're in. And then of course we do Kirby Bauer. That's the lab that we do. We get antibiotic discs, and we do the Kirby Bauer method. And then this year I added, they researched natural antimicrobials like lemon juice and garlic and cinnamon, and they made their own discs and had to do a gram-positive bacteria and a gram-negative, and they looked to see if their own disc worked, which the kids loved. They thought it was super fascinating. Did they all work? Absolutely not. And I think that's the beauty of this class, is that they actually never know if something's going to work or not. And this is probably one of those first classes they take that's not like a cookie cutter recipe, like add this, and then here's the prescribed outcome. They literally have no idea what's going to happen. And it's like real science at work, and it's beautiful. And so I love that I get to introduce kids to that idea. And that was a lot.
No, that was great. I mean, I wish I could go, in time and just take your class back in time. It sounds so interesting. Definitely, it's great, and yeah, I mean, microbiology, it's definitely not very linear, and a lot of times, there's a lot of gray, there's a lot of information. So yeah, it's not easy like following a recipe, even if you do that, right? Things don't work sometimes. It's very unexpected, and sometimes, organisms they have different morphologies. They, like we say, they don't read books, so they do what they want, and you see atypical presentations, atypical patterns. So, but it is so good to get exposed to these concepts, and especially the one with antibiotics that I think that's growing up, and then we don't think about this stuff, and maybe we're taking something and then we stop taking it, or you see people taking antibiotics for things that they don't need them, and when you start learning about all these things like AMR, and so it's really good to being exposed to all these concepts. I had a question. You mentioned yeast and the microscope. Do you do any type of gram staining?
So in our prokaryotic unit, so we have a unit that's called prokaryotic functional anatomy. And my goal of that unit is one, to teach all the different parts, right? Like so we're looking at like capsules and cell walls and flagella and endospores, right? We're doing all of that. And instead of teaching the lab technique by itself, I try to incorporate it into the lecture. So for example, we start the very first day of that unit with learning about cell shapes, cocci and spirilla and bacilli, but then like all the unique ones, right? Because there's so many that don't even fall into those categories. And I give them Play-Doh. So I give them Play-Doh and they have to model all of these different shapes with their Play-Doh. And it's on a Google Slides. So they make a little Play-Doh, whether it's like a diplococci or a streptobacillus or whatever it is, right? They make their shapes. And the next day in lab, they have 6 or 7 samples, whatever I'm able to get. I order through Carolina, so whatever I'm able to get from them. And they'll do like Micrococcus luteus is one, Bacillus megaterium, Bacillus subtilis we can use, E. coli. Um, there's a host of other ones. And what they do is they learn to simple stain. So they simple stain those bacteria, and then they match it to their Play-Doh model to see how we have this model and it's like perfect, but then what does it look like actually under the microscope? Which is very different, right? Like, and the kids are sometimes like, wait a second, I thought you said this was blah, 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 and it doesn't really look like that, or it's hard to find, or whatever it is, right? So that's how we start. Then we go on to cell walls and we diagram out gram positive and gram negative. They draw their parts, they label them. We talk about what peptidic glycan is and how it's put together. LPS, we do, the two membranes, all of these things that they don't have any clue about, right? Because they've never actually gone in depth onto a cell wall. We teach that in bio. We're like, hey, it has a cell wall. And then like, that's it. And so we go through a pretty big series of lectures for those two things. And then they gram stain. And so I give them a gram positive, I give them a gram negative. They gram stain those separately so they see what they're supposed to get, right? Like, and that takes, most kids take probably 3 or 4 times trying it, right? Just to even get it. Then I make them mix them together and do a gram stain so that they can see the purple and the pink together in their slide. And if they don't, then they know they've screwed up something, right? So then they can go back and restart. And so they do this. I mean, I bet we do it for 3 days because it's just hard, right? Like it's something that they've never done before. It's a lot of trial and error. They'll come up and be like, what did I do wrong? And, it's like, well, I don't know. I didn't watch you do I wasn't able to watch you do the whole thing. So I try to guide them in what I think they may have done. If there's, maybe you washed off too much with your alcohol, or maybe you didn't let your stain sit long enough. So I try to guide them in some things to try, and then they'll go back and try again. And then I assess them on this, which is the first time I think these kids have ever had a lab assessment. And I give them an unknown, and they have to tell me the shape and the correct gram stain for it. And sometimes it is crying, which is like not that big of a deal. I'm like, it's okay, like relax, like we're gonna get this. And they'll have to come back in sometimes after school and redo it. But that's the thing about science is like you can try again, right? Like if it didn't work in the lab, you go back the next day. It's not like you just automatically fail because you didn't do it correctly the first time. And And, I don't want them to feel that way. I want them to love micro. And so I always give them lots of opportunities to come back and try again if they feel like they can do it better. And so, yeah, we do gram stain. They love it. They will tell you that's probably the most favorite thing we do all year is they just love learning how to stain.
Yeah, I mean, Gramsing is definitely it's something that takes a lot of practice and repetition, and that's what we do as well when I'm teaching. And you give them feedback, and even when you're experienced and you've been doing it for many years, we still mess them up. But yeah, it's like you said, having them say, okay, yes, it's okay, you can just go ahead and repeat it because that's something that you do notice because the way that with science and when it comes to microbiology, the same thing that we talked about, it's not black and white. So you do see students that they get frustrated because they don't get it right the first time, which maybe they can do that in other classes and they ace their tests and they do really well and they get it very quick. But then almost all of a sudden they're like, oh, I can get this gram stain or I can get this and I'm struggling here and just, but it's okay. It's just that's what microbiology is. It's a lot of practice and repetition and you do it over and over again and that's how you get proficient. So that's really good. I like that yeah. At the very least, yeah, it's, like you're saying, it's okay. You can just repeat it and do it again, and that's good. Yeah, because that's something that maybe makes people stay away from micro, which is unfortunate because we do need microbiology. So I had a question. I know that when we were talking, via email, you talked about something, the great yogurt cook-off. Can you talk more about that, please?
Yeah. So, okay. So we have a microbial metabolism unit, and I can only imagine you're thinking, wow, that's so interesting. Kids probably love learning the Krebs cycle all over again and fermentation, all that stuff, right? Like everyone cringes. So they've had 2 years of doing cell respiration and fermentation and aerobic, anaerobic, and all that stuff. And so I try to be a little more application oriented with it. So we first start, I do a quick concept map over reviewing all the things that we've talked about. We go back over fermentation, we go back over Krebs, we talk about electron transport chain, all that oxidative phosphorylation, all that stuff, right? So we do a concept map. That's my lecture day. And then the next day, they actually begin a fermentation food research project and they make a presentation. So they try to find foods that are fermented that use microbes to create. And they create a presentation and they bring the food in. And so we have like kids get to snack on food and they present about how it's made and all this stuff, right? And that's fun. The kids like to do that and it's a good time. And if you bring food, everyone's happy, right? Like that's how you snag high schoolers. If you ever want to know how to do it, food is your answer. And then I give them this. I tell them, hey, we're gonna have the great yogurt cook off, and they break off into pairs, and they are responsible for finding their starter. So they can order it on Amazon. They can go to like a Whole Foods. They can just bring in a yogurt sample, right? Like you can just grab a Oikos or whatever the good yogurt, not Yoplait, it doesn't work, but something that has live cultures in it. They bring it in, they pick the milk that they want to use. They have to do a little research. I give them an article from like Good Housekeeping or something like that has how to make homemade yogurt. So they bring in whatever milk they want to try, and they bring in their pots and pans from home. We put their saucepan on the hot plate and we make yogurt. And the next day they bring in their mason jars. I put them in our incubator. We incubate it overnight, which makes very tart yogurt, by the way. It's a little bit of a long haul in the fermentation time, but then they come in and the kids set up their little tasting areas. They mix all their goodies into their yogurt. Sometimes they'll bring in like cookies and because they want to win or whatever. And the kids get to walk around and taste everyone's homemade yogurt. And I know a lot of people are going to hear this and be like, I don't know that I would ever eat something my kids made. And it's not for everybody, and that's okay. But I have now tasted probably 40 homemade lab yogurts, and I'm still here. So it can be done. And the kids love it because they are. So once they get all their taste testing right, they actually have to look at their yogurt under the microscope. So they take a smattering, put it on a slide. They can stain it or not stain it. Usually it doesn't matter. They put it under the microscope and then they have to find the bacteria that they thought they used in their starter. So a lot of the starters, they tell you exactly what to look for, lactobacillus or whatever it is. And so they look at that under the microscope and they have to draw me a picture and they have to label it, what type of bacteria did you find? And they have to go over like why their yogurt turned out well or didn't. So some of them have really runny yogurt, some of them have really, really tart yogurt, and so they have to explain the science behind how that yogurt was made. It's not all fun and games. They do have to give me a little science on there, but that is probably, besides gram staining, that's probably number two on my kids' favorite things that we do in class.
Oh, that is great. And before I move on to the next question, I just wanted to ask real quick, as you mentioned about the yeah, so they do the staining, and do they get to do any type of plating and streaking, or do they?
Yeah, so we actually do a lot more labs than what I've already said. So in the diversity lab, at the very beginning of the year, the very next thing we do is actually streak plating. They streak plant from soil samples. So they go out and get a soil sample. They do a cereal dilution on it, and then they streak plate it. And so then they have to do colony morphology. So I give them all the terms of margins and things like that. And they have to, look at their colonies and see what the margins are, elevation, size, that whole bit. So they do streak plating. They do capsule staining in our prokaryotic unit. I teach them to capsule stain, which, let me tell you, is 98% of the time a disaster, but it's a really fun stain. And I switched to nigrosin this year, which really helped. I was using Congo Red because we only have access to so much stuff. So that's kind of the difference between teaching high school and college, is like I only have so much access to certain bacteria because it's a safety one. I only have so much access to certain chemicals, things like that. So we do capsule staining, which they also love. They do endospore staining with malachite green. And they think that's the coolest stain ever too, because you get to just run the green over, your paper towel, and it just drips everywhere and it makes a massive mess. It's probably my least favorite one to clean up after. But they also very much enjoy that. We do carbohydrate fermentation tubes with phenol red broth, which when it changes color the next day, and that's in our metabolism unit. We actually do that after the yogurt. We'll actually do some actual labs for it. And then in that same unit, we do oxygen. So they make TGB tubes, and then we grow anaerobic and aerobic Clostridium species and things like that in the TGB tubes. So that's pretty fun. I'm trying to think of what else. We have a gel electrophoresis lab coming up in our virus unit that we get from Micro PCR company, which the kids really like. We also do the yeast one where they have to feed the yeast different things, and then they look at budding rates. They look at it in a microscope, and they have to find the budding yeast, and they feed them different amounts of things, or they add things, they heat it up or cool it down, and they have to see how that affects reproduction. So we do. I try to make a list of all the labs because I can't even remember half the ones that we do, but we do so many because that truly is the most engaging thing for these kids. And to be quite honest, I think it gives them a leg up in college. I had a girl write me an email this couple months ago who's in college right now in micro. And she was like, Miss Townsend, we just did gram staining. I was the only one who knew how to do it. I was so proud of myself. I felt so confident going into class. And like, let me tell you, as a teacher, like that's all you can, right? Like that's what you live for, right? Like you live for that email. And so I have incorporated more and more labs every year because I know when they get to college, that will give them more confidence, that they will go in and feel really good about what they already know how to do. And so yeah, I do a lot of labs. So if you have questions on those. We also incorporated case studies this year. And if we have time, I'd love to hit on a few of those things as well.
Yeah, please. Yeah, and what you said definitely it gives them a leg up. I mean, it's so much information that they go to take a college class already understanding how, gram stain and some techniques and understanding some basic microbiology concepts. They definitely are way better off than their peers that haven't. It's a lot of information, and it can be daunting sometimes. And it's something that is not for everyone, and maybe some people struggle in school with it, and it's so much. But yeah, definitely, I love to hear about your case studies, please.
So this year, in my school, we have to do a research question. Teachers do. It's how we get evaluated. We pick a research question. And so my research question for me this year teaching was, how can I bring more real world content into my class? Like, how can I, we teach about diseases, we look at them, but like, how can I make it, I don't know, just how can I make them like physically, latch onto it, right? And so I found these books on, I was actually trying to make a list of what they were so I could tell you about them. I'll find them in my notes list here, but we have all of these case studies. I get them from, okay, there. I get them from, it's called Case Files Microbiology and Problem-Based Microbiology. And within these books, they have patient case files. And so they'll give you a description of the patient. It'll say like patient 41 and comes in with blah, 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 and it lists all their symptoms. If they have a fever, it will list all their lab info. So like what their white blood cell count is, any lab work that they did, blood pressure, that sort of thing. And what I do is at the beginning of the year, I give the kids a norms value table. So what the normal lab should look like, because they have no classes that they've done this before. So they have to have some sort of reference point, right? And when we get in our immune system unit, that's why I moved my immune system unit to the beginning of the year, because I wanted to be able to go like, what does a white blood count mean? What are the white blood cells? Like, what are like, right? Like, what are all these things? And so that's why I moved that up. So they have this norms value table, they get their patient file, and the first thing they do is they have to go through their patient file and find all of the things that stick out that look abnormal. And they have to tell me what they think it means. What does the fever mean? What does a high neutrophil count mean? What does a high macrophage count mean? What does a low red blood cell count? I don't know, whatever it is, right? And so they annotate their case, and they do this in groups. It's very important for me that they work in groups. Working by themselves, I feel like kids really shut down. So they're in groups of 2 or 3 all year for everything we do for the most part. And I actually use this as their test grades a lot of the times too. It really tells me what they're learning. And so they go through and they annotate all of these case files, and then I give them leading questions. So in the books, it gives you the answer. It says like, this is what the pathogen is, or this is what the organism is. But they can't get that page until they've shown me what their next steps are. So they annotate all their symptoms, tell me what they think is wrong, which we've learned about all these bacteria in the unit. So they have some idea of where to start. And I say, well, what would you do next? If this was your patient, what would you order? What would you have them do? Would you take a fecal sample? Would you do a urine sample? Would you do a nose swab? What are you looking for? And if they can tell me that, and if they tell me what they think it is, give me a list of possibilities, then I give them the next page to the case study. So it's like kind of, I try to set it up as real world, like patient enters, you think you know what it is, you order the test, then I give them the next page that has the results of the test that they wanted to order, and then they go through the rest of the case study and they figure out what it is. And then a lot of times the case studies have a lot of information about how it was treated. So they have questions they have to answer, like, how was this treated? What antibiotic was used? Was it antibiotic used at all? Was it a virus? How could you prevent this happening in the future? And I took a little survey at the end of my first semester about how kids were feeling about these case studies, and overwhelmingly, they loved them. They felt like they could relate the information they learned in class to how it could be used outside of class. A lot of these kids have doctors as their parents. And so one kid was like, man, I heard my mom on the phone the other night, and I actually knew everything she was talking about because I had heard all those words before. And so, just giving them this sense of real world application. So for example, when we're talking about, capsules. When we're talking about capsules, we'll talk about Klebsiella pneumoniae. And so that's one of their possible case studies that they could get. So we go through what Klebsiella is, how the capsule's related to its like virulence, that sort of thing. We do Borrelia when we talk about axial filaments. We talk about E. coli when we talk about cell walls. We do prokaryote. We don't just do prokaryotes. We do protozoas. We do leishmaniasis, Giardia lamblia. We do amoebas. And so while they're learning and looking at all these things under the microscope, they're also learning about the disease. And then they get a case study at the end of the unit. And like for the protozoas, I actually gave them the patient file, but then they got their lab results in, but it was under the microscope. So they had to go to the lab, look at their results under the microscope, which was just a slide that I already had, and I sort of taped up the answer. And then they had to tell me, they had to diagnose their patient. And so I think there's a real need for case studies, but I also think it's a really easy thing to incorporate it. You just incorporate it right along as you're going. When you're talking about the immune system and lymphocytes, T cells and B cells, throw out Epstein-Barr virus, right? Like, let's talk about mononucleosis. Like, you have all of these really easy pockets that you can throw these case studies into as you're teaching. You don't have to save it for like a disease unit at the end. Because if you try, you'll run out of time because micro takes forever to teach. So anyway, so I just love those. That's probably one of my favorite things I've incorporated this year.
Wow, you're teaching so much, and like they're definitely learning a lot. Um, yeah, and with case studies, I think it's easier too, as you're learning the material, right? To put a visual on it and start thinking more, and definitely helps, helps with learning. Um, so as far as right with any sort of a class, there's a way to assess the learning, and it'll be like an exam. Or when it comes to micro, we tend to do like practicals or unknowns, like you mentioned. Um, and then so you give them projects. So how do you, assess their learning and grade them? What do you do?
So this is I like to call it a non-traditional class. So these kids have come from all these AP classes, right? Where they are multiple choice FRQs, multiple choice FRQs. Here's your grade. And I just feel like the stress of that is starting to wear on some of these kids. And so I decided I wanted to make this a non-traditional class. And so the assessments really are varied all year long. So for example, in our immune system unit, we learn everything we need to learn, right? We get to the end and I give them a scenario and I give them a big giant piece of butcher paper, like poster board. And the scenario is like, Katie was walking on the trail and she cut her leg. Explain, she starts feeling sick the next day. She has a fever, blah, 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 whatever it is. And I make them literally make a giant concept map of how she got sick and the way that the bacteria or virus or whatever it is whatever the prompt they get, how it made its way through the body and how the immune system fought it off. And so it tells me really quickly whether they know stuff or not. They get their prompt and they have to work. They get to work in a group, so it's a lot of the pressure is a little lower, but it's not traditional. They don't get a multiple choice test that says, what white blood cell is the first one to, find the pathogen or whatever it is. It's just not written that way. But when they get their prompt, they really need to say, well, first, it broke the first line of defense. The first line of defense is her skin. It broke through that. Neutrophils would have been the first ones to act. Chemical messengers were released. A fever was, that sort of thing. And so I get a pretty good idea pretty quick, like if they know what they're doing or not. But that's one way I assess is they have some sort of like group, concept map or open-ended thing. And honestly, all my kids went such a different route, how they drew it out. Some of them wrote paragraphs, some of them drew pictures, some of them did concept maps. And so I like that open-endedness for them. We do a lot of practicals. Probably the biggest practical we do is in our prokaryotic unit. They get a mixed tube. So they get, it's a gram positive and a gram negative. Every group gets a different set of bacteria. And they have to straight plate to separate the colonies. They have to simple stain, capsule stain, gram stain, and endospore stain all of their samples, and they make a presentation about what bacteria. They don't have to name the bacteria because they haven't learned enough to do that. They don't have to say, oh, it's E. coli or it's whatever. But they do have to give me the shape, and they do have to correctly identify the gram stain, the capsule, and the endospore. And so it's a lot of pressure. They have a whole week, they have about 6 days to do it. And that's their assessment. Can you do the stains? Can you tell me what they are? A lot of it's processed. So the lowest grade they can get on it is a 70. Like, even if you got all of them wrong, it's about like learning in the process, and you know what I'm saying? So I try not to, I don't ding them a million points because they got it wrong. It's just, where did you mess up? They even have a little part on their Google slide. If they do mess up, they can go back and say what they think they did wrong, that sort of thing. What was the human error? So they have some leeway in that. We do a lot of practicals. We did the project for antibiotic resistance. That was the little coloring or children's book. They did, presentations. I had them research different types of fungal infections, yeast infections for the most part, and mold. And they presented about their research on that one. We've taken maybe one multiple choice test. That's really not. We just really don't do that, and I think the kids appreciate it. And like a kid told me, someone asked me about why we don't do as many, and I kind of told him why, and he was like, I feel like I've learned so much in this class, though. It doesn't matter if I didn't have that, because I feel like I have learned more in this one science class than I've learned in many of my classes, because it's just the nature of the class, right? Like if you have it, if it's engaging, if kids show up, if they want to be there, if they're excited about the content, then they're going to learn stuff, whether you multiple choice test them at the end or not. So a lot of their labs go in as grades. Most of the labs I do take as a grade, but I only take about 8 grades in 9 weeks. So that may be a little different than your typical traditional class. We don't have a lot of daily grades and things like that.
Okay. And so now that you mention books, right? So I see that you actually created a coloring book. Can you tell the audience more about that?
Yeah, so it's funny because it really, I honestly didn't think this was going to happen. It was sort of born out of boredom, maybe. I don't know. Me and my kid, I have a 14-year-old boy who's also really into science, and he's also a very good artist. And we were just sitting there talking one day, and I was like, hey, it'd be cool if we like wrote a kid's book on microbiology. And he was like, really? What like would it be cool? And I was like, yeah, like we could do like the ABCs of micro, and I could do all the writing and maybe come up with like the things, and then you could draw the pictures for me. And I, really it came out of a way of like bond with him, of like having this project that we like put together, him and I. And so so we did it. So I gathered all my words, I got on Canva, I, made all my formatted everything. I tried to write the stories about each thing in an easy to read format for younger kids to get them interested. And then I would give him kind of the basis of what it is, and I'd show him some sketches and some diagrams. So because he's never taken microbiology, and so he doesn't necessarily know what everything is. And then I let him kind of just do what he wanted to do. And then we published it on Amazon. And so there it is. It's pretty neat. We've actually sold like 60 copies. So, which I think he thought we might sell like 5, to like my mom. So it's pretty awesome.
Wow, yeah, that's definitely, yeah, 60 copies. Wow. So then, as you said, it is in they can find it in Amazon.
Yes, it's called ABCs of Microbiology, a coloring book for future science nerds. And yeah, it's on Amazon right now. I hope to, I have a website, or I have a blog. I will tell you this, I don't pay for it. It's like a wix.com, address. It's called Mrs. Townsend Teaches. And one day maybe I'll pay for the full thing, and I can sell digital downloads on there. That's kind of the hope for the future. I think he wants to do another one that's just the ABCs of biology, and we'll just, keep playing off of it and make it a series and things like that. Maybe that'll be a project for this summer. But yeah, it's on Amazon. It's there. You can also search my name is Meredith Townsend as the author. And so anyway, it's pretty exciting. So, and also, if you need free lessons, if you can find my blog out there, Mrs. Townsend Teaches, it's a wix.com one. I have free lessons for micro and biology on there, things that we've done. So I'd love for anyone who just needs a little help or ideas to go out there and hit it up.
Yeah, so I'll go ahead and I'll put the link on the show notes to your blog. And when I'm doing the intro of the episode, I'll go ahead and also mention it. Um, okay, so you do have like some future plans of maybe doing something else, and that's great. That's something, right? Like a project like that, and maybe can you talk also a little bit about your blog if you can? I know you mentioned it sometimes on X when I see your posts and stuff. So do you like post regularly, or what do you normally? You said that they can find like lessons. What else can they find?
Yeah, so I used to post regularly. You know how life gets you sometimes. I posted quite regularly up until about 2023. And then last year just fell off. Everything got busy. And, I have a 15, a 14-year-old, and a 12-year-old or 11-year-old. And so we're just, it's a little chaotic sometimes. But I've kind of reawakened it, right? And so I've been posting, trying to post regularly the last couple of weeks and bring it back to life. But again, it's like, if you're in biology and you're stuck trying to teach biomolecules and you don't know how because it's boring, um, that's okay. I have like a 5-part like series on how we teach biomolecules, and I literally go over like how do I teach carbs, how do I teach lipids, how do I teach nucleic acids. It's got worksheets and activities. Well, maybe not very many worksheets. We don't really do a lot of worksheets, but it's got um, YouTube videos that I like to use, the project that we do, how to teach dehydration synthesis through a game, right? Like it's literally links to my Google Drive. It's view only copies, and you can just go and make a copy for yourself and use it like you want to. But it's got some microbiology lessons. Like I think my diversity lab is on there. Um, I think there's a microscope lab on there, a size comparison lab where they basically blow everything up 1,000 times, I think, and then they draw it out and they compare the sizes of like a hair and a bacteria and a prokaryote or a protist and things like that. Um, anyway, there's all sorts of things. I've got some immune system lessons up there that I use, one of the labs that we do. So if there's something you need, it's probably on there. And if not, my contact info is on there too. I'm happy to write blog posts if someone's stuck on an idea or anything like that. I've got about 16 years worth. So
Well, before we get rid, we're getting ready to wrap the episode, but is there anything else that you want to add?
no, but if you're just a microbiology teacher out there and you're looking for a space, I'll give my info and he can put it in the show notes or whatever. Please reach out. We have a Google group. We'd love for you to join. We have a shared Google Drive. I I basically just copy and paste all my lessons into it, and you're welcome to use whatever you need to get through it. Teaching micro in high school is hard. It has its setbacks, but it's also extremely rewarding. So the more teachers we can get and help out, the better I think we're going to be in the future, because we are truly educating kids who are going to be stuck with solving our problems, right? Like they're going to be the ones to solve antibiotic resistance, because we're not going to have a choice but to get these kids in there in 10, 20 years. Who we teach is going to be the ones saving us, I think. I truly believe that. And so if there's anything I can do to help inspire or even just get you through the muddling of the waters, right? Teaching high school micro, I'm happy to do so. Because it's just the more we have, the stronger we are as a group. So it's a great group. We've got quite a few really just smart teachers in that group who, one in particular has helped me a ton. I was struggling with capsule staining this year. She has a method that she felt like worked really well. I reached out to her. She emailed me all her stuff, told me what she thought I could do to fix the problem, and it did. And so, we're very there. We're here for collaboration. We're here to make sure that everyone is being their best selves. And so I would love for you to reach out to me if you want to join.
Definitely, such great words, and I'll put the information in the show notes if you can send it to me. Um, but yeah, no, definitely, it's it's so great, and I know you sound so passionate and that you love what you're doing, and it's I don't know, so great and refreshing to see someone that loves teaching that much, and you're doing such a great job and doing so much. So definitely, thank you for all you do and helping these kids, learn microbiology. And like you said, right there in the future, it's might be up to some of them to solve some big problems. So you're laying a foundation there that it's very important. So thank you for the work you do. And it's such a pleasure to know that you're doing this type of work, and thank you so much for taking the time to being in Let's Talk Micro.
Absolutely. I appreciate you having me. Uh, Teaching is awesome. Let's get some more teachers out there. Right? Like I think there's a lot of negativity around public education, and it doesn't have to be that way, right? It doesn't have to. You can have a positive work environment. You can love kids, and you just got to find a school that fits you. And so I appreciate you having me on so I can help spread that message.
My pleasure. And like you said, yes, teaching is very, very rewarding, and it's something that you need to have the patience and the motivation to do it and the dedication, and it's a big responsibility, but one that you really do well. So once again, thank you so much. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about teaching microbiology in high school, right? Those of you that love micro, I bet that you're finding this so amazing that this opportunity is being given to students out there. I don't know. I'm so excited to sharing this with you. As always, please continue bringing that passion to what you do. It's so important. You do such great work. And thank you so much. I started this podcast to share information, to learn, because I have learned so much by preparing for it, meeting guests, learning from their expertise. So it has been such a great journey. So one that I have enjoyed so much. So thank you for the 100,000 downloads. Thank you. Again, thank you. So grateful. As always, stay tuned. Great things coming your way. So stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.